Hello again. Today we're going to talk about replication, and if we talk about replication, there are two topics that we're going to see in these lectures. One is the proof to show that replication occurs in semi-conservative manner, and the other one is the replication process. Now, previously, before they understood that, before they have proof to show that replication occurs in a semi-conservative manner, there were three different hypotheses. One says it's conservative, the other one conservative. That means whenever you have a new strand, both of the original strand remains, right? And then there's dispersive. Dispersive is just a mixture of the new strand, and the old strand is just a mixture of both new and old DNA strands. And then you have semi-conservative. By virtue of its word, by virtue of the meaning of semi-conservative, that means half of the parent molecule is conserved. So in this case, from yellow, the black is the new one, is the newly synthesized strands. So on the first generation, you have a mixture of yellow and black, and then on the following generation, again you have the yellow and the new black, and then the initial black now becomes a template and you get a new black one, and so on. So that is semi-conservative. Now don't get mistaken to think that there are three modes of replication. No, that's not correct. It is thought. These are different ideas that people had at that time in terms of trying to explain how the replication works. Now, Meselson and Stahl went on to prove that it is replication occurs by semi-conservative, and he was able to do this based on two important factors. One is that his ability to synthesize, to allow the bacteria to grow in different medium, so that when bacteria grows, it makes new DNA. So if the DNA is given a DNA with made up of the 14N isotope, so all the DNA will have 14 Ns, and then if you provide the bacteria with 15 Ns, then you have two different types of DNA molecules in two different cultures. And the other important factor is this slide here. This is called density gradient centrifugation. What you do? I'm sure you know what centrifugation is. So density gradient, that means in the tube itself you have a medium. In this case is cesium chloride, in which it has undergone a few days of centrifugation, just the medium, so that what you have is a gradient density. The top is not very dense, but the bottom is very, very dense. That is depicted by the dots. So if you have these two molecules, two types of DNA molecules, 14 and 15, and you can mix them together, and then if you centrifuge, you actually get separation based on the density. So these two factors, the ability to synthesize different types of DNA bearing different nitrogen isotopes, and the ability to separate, has allowed Meselson and Stahl to provide proof for semi-conservative replication. This slide shows the theoretical expectation, right? So that means to say, you have these two different molecules, 14N and 15N. If it undergoes through conservative replication, I'm sorry this is in Malay, but but I think you can understand, for your internationals, you can understand what it means. The hypothesis says you start off with 15N, but in the second generation you have 15N and 14N, and therefore you get two bands when you centrifuge and then a density gradient. And then the following cycle, replication cycle, you get 3 to 4, so that's why you get the thickening of that band. And then for dispersive, it's not easy to really explain because it's quite random. So you start off with the 15, and then you have a mixture that goes in the middle, because it's a hybrid. It's neither 14 nor 15, it's a mixture, so you get a band in the middle. And then this one continues as more and more 14 becomes available. All right. Whereas in the semi-conservative, you start off with 15N, and then in the first generation, because it's semi-conservative, that means you get a hybrid molecule. So each molecule has one 15 strand and then one 14 strand, and because they are the same, and that's why it remains in the middle. Now this is the defining step in the second replication generation. So that white becomes the parent, the synthesis of a new red. That red becomes the parent for the synthesis of new red. That white again will give you this molecule here, and that red will give you this new molecule. Again, semi-conservative replication. And because you'll see that you have 14 ends and you have a hybrid. So this is 14 ends, and then this is the hybrid. And for every subsequent generation, you still have the hybrid and then a lot more 14 ends. That's why it's thickening. This is hypothesis. So then you do the experiment. How did he do experiment? He allowed Meselson and Stahl allowed the bacteria to grow in a medium containing 15 ends. Right? It allows that to continue to replicate or to grow. And then what he will do then, he will take an aliquot, a portion of the bacteria, and then grow them in the presence of 14 ends. Bacteria will grow, bacteria will divide, and then in the process will make new DNA. But now the bacteria will make DNA based on whatever is available, which is a 14N. It starts making new strands. So in his experiment, right, so this is the actual experiment, this is just the abbreviated result. So he took the aliquots, or I think every 10 minutes or so, then he gets this kind of results. Now, because at that time they don't know how long the bacteria takes to divide, so that's why you have 0.3 generation, 0.7. So you can remove all that, and then you just take here 0 generation, first generation, second generation, third generation, and fourth generation. And you can see the outcome of the experiment mimics the semi-conservative replication. You have 1, 15, you have 1 in the middle, and then you have 15 in the middle, and then 14, and then it goes on. So this is what you see in the experiment. Here is the 15, here is the middle, here is the 14, and then the 14 gets thicker and thicker, and this is control. So from this experiment, he was able to show that DNA what is indeed replicated in a semi-conservative manner. Alright, see you in the next video.